My name is James Kim, and I am one of HESI's scientific managers. So it's my pleasure today to introduce you to our kind of spring outreach webinar. I think that most of you are familiar with HESI, but I thought I would just touch base on what HESI is. As you know, HESI is a nonprofit research institute with a mission to engage scientists from academia, government, and industry to identify and resolve global health and environmental issues. This is to support HESI's vision, which is to create science-based solutions for a sustainable, healthier world. I guess before we get too far into it, can everybody see the slides okay and hear me okay?
Yes.
Okay.
Okay.
Great. So one of the committees that I manage is the DART committee, which is the Developmental and Reproductive Toxicology Committee. The reason why I'm presenting this today is because the presentation that you're about to hear is focused on developmental toxicology. And the DART committee has been keenly interested in the use of zebrafish for developmental toxicology. The DART committee currently has 23 sponsors and a number of government agencies that are involved in its projects. As you can see, the sponsoring companies cover a wide range from pharmaceutical companies to chemical companies and CROs, as well as the biotech companies. This slide here presents the leadership of the DART committee. It's currently chaired by Bruce Beyer of Sanofi and co-chaired by Jane Stewart at AstraZeneca. We have a number of advisors representing the EPA and the FDA, as well as the RIBM in the Netherlands. This is the Developmental and Reproductive Toxicology Committee's current project portfolio, and I wanted to show this primarily to draw your attention to several projects that are ongoing that are related to the presentation today. The presentation today is about zebrafish, and one of DART's ongoing projects has been in alternative assays, which you can see here. The DART committee has organized several workshops in this area, the last one being in 2009. We also have a project here called the Consensus List of Developmental Toxicants. This is actually a list that we're putting together for use by assay developers to test their technology for developmental endpoints. We hope to have publication of this list later this summer. Our testicular toxicity project is also focused actually on in vitro testicular models. We had 2 workshops last year, and we should have a publication from that workshop probably by the end of the summer. Another major project that the DART committee is undertaking is what we're calling the rodent versus non-rodent second species. I think for those of you in the pharmaceutical sector have been aware of some of the issues and the discussion about using rabbit as a second species for testing for developmental toxicology, and the discussions around whether or not testing in that species could be delayed or even not followed through with if you use in vitro technologies. That's kind of a discussion that's been ongoing, and the DART committee has decided that in order to really understand what you would do by delaying testing in the rabbit, we're actually going to create a database and collect information comparing the rat and the rabbit developmental toxicology testing from pharmaceutical companies. So we're collecting that data probably this summer, And I think that those of you on the phone that are HESI members, you probably will receive a survey, so I would encourage you to participate in this project by submitting your data. I think that it could be hugely important for the field. So again, just to briefly summarize, the DART committee held an alternative assays workshop back in 2007. They followed that up with a workshop in 2009 on alternative assays. There have been other workshops that the committee has held related to the use of in vitro technologies. There was the Developmental Toxicity New Directions workshop in 2009. As I mentioned earlier, there was the testicular tox in vitro models last year, and we also held a workshop on the consensus list of developmental toxicants last year. So that brings me to the end of the introduction of the DART committee and its interest in this presentation today. It's my pleasure to welcome Karen Augustine from Bristol Myers Squibb. She'll be presenting an assessment of the zebrafish model for developmental toxicology screening. Karen received her PhD in cell biology and anatomy from University of North Carolina. She was the head of molecular teratology at GlaxoSmithKline from 1997 to 2002. She then became the associate director of reproductive toxicology at Bristol Myers Squibb in 2003 and is currently a research fellow in discovery toxicology at Bristol Myers Squibb. She oversees reproductive toxicology testing in discovery and preclinical development. She's been active in a number of HESI activities, including the DART committee. She has also served on the Emerging Issues Steering Committee and was actually the co-chair of a recent HESI workshop on epigenetics. So with that, I will turn the presentation over to Karen. We will have time following her presentation for questions. If you look on your the webinar software to the right, you should be able to right-click your name and or signal your hand, and then we can take some questions that way. So Karen, I will turn it over to you, and I'm right now on your title screen.
Okay, well, thanks very much, Jim, and thank you all for joining this webinar tonight. I really appreciate you spending the time and the interest in learning about what this consortium has been up to, and konnichiwa to all the Japanese who have joined the webinar. So I'm going to move to the next slide. This project that I'm going to describe is a cross-pharma consortium effort. The history of this was the consortium was put together back in, I guess, around 2009, and it was started by Anne-Lee Gustafsson, my colleague from AstraZeneca. She had given me a call. She's out of Sweden, and she had given me a call and, oh, I'm going to be visiting the U.S. on the FDA. Would you mind if I stop by and chat with you? And came up to New Jersey, where I work, put her around what we did, and told me about this dream she had of trying to get people together to work on really evaluating the developmental tox assay very carefully with a large set of pharmaceutical compounds. And my group had worked with zebrafish for quite a number of years, so he had asked me if we could partner together and try to get this off the ground. So now we have about 5 pharmaceutical companies involved in this effort, with quite a number of people that are listed on this second slide, and they've all been really amazing contributors. We also with a small biotech company that helped us executing some of the fish experiments, and that's Lampire Biologics. Okay, next slide. So our overarching goal for the consortium effort is to establish a harmonized zebrafish developmental tox assay that's been rigorously tested for performance. So in other words, what we wanted to do is many pharmaceutical companies are currently involved with trying to find ways to proactively test drugs in the discovery for different types of safety liabilities. And the thought was, well, it looked like zebrafish was potentially a promising cell model screen. So if we could all work together to really rigorously test it and get a protocol together, then if we start to run the same type of assays early phases, and this type of data is shared with regulatory agencies, they'll be secure that the experiments were carried out in the same type of way. And part of the goal of this consortium is to be very open about what we learn along the way during the project, which includes communicating publications as well as specific forums such as HESI. So we all learn along the way the positive attributes of the assay as well as the potential negative sides and the gaps with the assay. Let's move to the next slide. So the consortium activities have been divided into two phases. And we've recently completed phase one, and that's most of what I'm going to be talking about today is what we have learned in phase one. And phase one included running some pilot studies to just make some decisions whether we would want to develop a zebrafish assay that keeps the chorion on or chorion off. Chorion is it's like a shell. It's a shell around the embryo. It's a protective shell that protects the fish embryo from different types of environmental results. But there was some concern that maybe it would actually be a barrier during compounds from being taken into the embryo. So some pilot studies were taken by a number of the consortium to see whether or not chorion really seemed to be a relevant barrier. And we made decisions there where we felt that we could we could move forward with an assay that had that kept the chorion on. That was significant from the standpoint of future goals of working with an assay that could be potentially conducive to high throughput screening, because to remove the chorion by standard methodology requires a lot of microdissection techniques. We worked together to put together a set of compounds that would be used, and we developed a protocol. And we ran that protocol in a form of a blinded inter-laboratory, and then moved on to doing some optimization studies and rerunning an optimized protocol. And in the second phase of the work, which is currently active, took the optimized protocol. We tested 38 proprietary compounds at 2 different laboratories, and that's the work that we're completing currently. So we'll move to the next slide. So let me start from, let's talk about what a zebrafish is as an organism. And zebrafish is essentially a minnow. It's a tropical minnow that is found in Asia, particularly in northern India in the rivers. And it has been used for many years in different types of research. For, I would say, 30 years it's been used in ecology testing, and it was a real staple of development genetics. Its genome has been sequenced and well annotated. It's conducive to insertional mutagenesis approaches that can link mutation back to the phenotype, and so it's favored by geneticists for quite some time. Next slide. In terms, then thinking about adapting this model for the use in laboratory screening, it seemed to be a very positive model. For one, it aligns well in context of the 3 R's. So if you're looking at a screening assay as a form of a animal alternative or replacement, The zebrafish embryo can fit the bill quite nicely because the parents are not euthanized in terms of harvesting the embryos. The husbandry is quite inexpensive. Assay costs are also very inexpensive. And the embryos and larvae can be cultured quite easily in multi-well tissue culture plates. Organogenesis is completed within only 72 hours post-fertilization. So what's beautiful about that is that it can very nicely model what teratologists want to see in terms of vivo studies. So compounds can be administered at a stage that's very similar to the developmental stage that represents the post-implantation mammalian embryo in embryo-filled studies. And then organogenesis can occur within a couple days and can be observed in real time that's occurring. There's a lot of cross-species conservation of developmental processes and pathways. I believe it's between 75 and 85% of most molecular pathways between zebrafish and And as I mentioned before, in an extensive evaluation of genotype to phenotype with databases that contain this type of information. So the model itself could be applied nicely to evaluate teratogenic mechanisms. Next slide. So in terms of thinking about this for the zebrafish for actually putting screen, the goal would be to use the zebrafish for safety screening, proactive identification of hazards. It would be a medium throughput assay, but possibly be applied to an automated platform, higher throughput in the future. In terms of setting benchmarks for performance, compounds would be selected that have definitive in vivo teratogenic information. In other words, these would be compounds that have been tested in multiple animals, test mammals, so that we knew whether or not they were definitive teratogens or non-teratogens. But looking at the zebrafish type of screening, you also have to consider up front there could be inherent limitations of a zebrafish assay. One is that there's no placental barrier associated with zebrafish. And so compound uptake is going to be very different than what is seen in a mammalian embryo fetus. There's also well-published known variations in compound uptake that aren't completely well understood at this point. And compounds that are insoluble in nature may be limited in terms of being able to be tested in this assay because the culture media is essentially very aqueous in nature, so precipitation could be problematic with some insoluble compounds, which would limit the ability to interpret data. And other approaches could be done with a modified assay that would involve direct microinjection of compounds into the embryo or into the embryo's yolk ball as an alternative. These are thoughts that the consortium might take on at a later time, but at this period of time, we were looking at a standard type of assay that would require, that would involve dosing directly to a culture medium. Next slide. So, as I mentioned, the assay is pretty simple in nature. Following a acclimated light cycle and feeding cycle, embryos can be produced quite readily on a schedule with a certain type of feeding regimen. And those embryos can be collected and then just plated in tissue culture plates. And in the consortium hands, we work with 24-well tissue culture plates. And these embryos are placed in a media called fish media that would contain drug, and they would be incubated for a period of time at 28 centigrade. And as mentioned, these embryos will continue to gestate along their normal time course. And so over a period of a few hours to days, one could observe the embryo's development in real time and then assess for morphology and viability at designated periods. Next slide. So what we decided to do in terms of developing an initial protocol was to work with some of the framework of what my group had published back a couple of years ago with a dechlorinated zebrafish developmental tox assay. That assay was used as the basic bones for the framework of the protocol. And the big drive for that was that we had promising results with the dechlorinated assay, but we also had developed a morphological score system and had published essentially a score guide that is a very good training reference, because our intention was to run an inter-lab assessment. So we wanted to make sure that all the different laboratories knew exactly how to evaluate respective structures and organ systems in the embryo and do it in a very harmonized, methodical way. So we decided to go with that score system. And what's pictured here in this slide is the day 5 post-fertilization zebrafish embryo. And that is the stage that we run our viability and morphological assessments, and this is done in the consortium work as well. There's a whole battery of structural endpoints that are evaluated, as well as growth measurements that are listed on the slide. Next slide. And then for each structure, there's a score that's assigned. The scores are on a 1 to 5 scale. 5 is assigned for a completely morphologically normal structure. If there's a subtle variation or anomaly that's considered potentially recoverable, that structure is assigned a score of a 4. And then if there's a definitive malformation associated with the structure, and that would mean something that would be considered non-recoverable in nature, that structure would receive a score of a 3, 2, or 1, the severity of the malformation. And a 0.5 is assigned in the case for a structure that does not appear to be evident. So this next slide is just providing some representative pictures of types of scores we would assign for different types of dysmorphology. Slide B, for instance, it's a picture of an embryo that was treated with an endothelin receptor antagonist, and I would think some people on this webinar would know endothelins are very important in neural crest development, and when that pathway is altered, first pharyngeal arch syndrome is a type of phenotype that occurs with loss of endothelin signaling. And this zebrafish embryo is presenting those types of dysmorphologies with a lower jaw defect, as well as some effects in the pharyngeal arch region. And they would be considered, in terms of severity, pretty moderate in nature, so that this combined jaw arch score would be assigned a score of a 2. In another case, the embryo in plate B was treated with I believe it was a cannabinoid receptor antagonist, which was teratogenic in vivo. It caused a lot of severe craniofacial malformations, and that also was recapitulated in the zebrafish embryo. And these various structures would receive scores of 2 or 1, meaning they're moderate or severe malformations, depending on the criteria that's associated with the score lexicon. Next slide. So the way compounds are classified for teratogenic viability uses a teratogenic index or a ratio. This is a ratio measurement of viability endpoint divided by the no adverse effect concentration. We found the concentration that causes 25% lethality divided by what is designated as the no adverse effect concentration provide a good set of values that we could use to set cutoff values that best classified teratogens from true teratogens from true non-teratogens. And so using this approach, ratios that were greater than or equal to 10 were predicted as teratogens, and ratios that were less than 10 were predicted as non-teratogens. From previous experience, what we also found was if we did not see any adverse signal through the highest concentration that we tested, the ratio would be considered undetermined, and we would default that undetermined ratio as a classification as a non-teratogen. And with our training set, we only found a 10% false negative error rate by using that type of criteria. So we used the same type of criteria with our initial consortium work. So moving to phase one of the consortium efforts, the members of the consortium picked 20 non-proprietary compounds for the initial analysis. And there were 10 definitive teratogens and 10 definitive non-teratogens in that training set. They were provided to 4 different laboratories with blinded IDs by another consortium member that handled all the compounds. And those compounds were run at 4 separate labs, and each lab, and there were 3 different types of wild types that were used in the assessment. So it included pond fish wild types that labs A and B used, and then the wick strain at lab C, and a proprietary fish in lab D. There was strict criteria for accepting data. For instance, we have found by running historical control analysis of experimental data from various labs that the background malformation rate should be less than 16%. So if we saw background malformation rates above 16%, then you would feel that experiment would fail the criteria for being acceptable to include in the consortium data set. Also, lethality should be less than 10%. So if there was a higher lethality rate, the experiment was not accepted. And also, all experimental and formulation procedures had to be in alignment with the protocol so that everything was done the same between the 4. So the study design included a concentration range that went from 0.1 micromolar through 1,000 micromolar. The DMSO was used as a vehicle in these studies. That concentration was either at 0.5%, or in some cases we did something called a double dosing to achieve some of these higher concentrations. So the background DMSO concentration was 1% in those cases. There was pH adjustment work that was done during formulation to optimize compound solubility, and then the working concentrations were pH adjusted to ensure that they remained within an acceptable pH range for culturing embryos, normal morphology embryos, I should say. And 12 embryos were tested for each drug concentration. 1 embryo would be plated per each well. And again, we used the 24-well plate format. So at 5 days after fertilization, we conducted viabilities, as well as we used the random selection method and selected 8 of those 12 embryos for morphological score analysis. Next slide. So with the compounds that we selected, they're listed in this slide here. And what I wanted to point out was there was a pretty good chemical diversity with these respective definitive non-teratogens. I mean, from the pharmacology side, they represent a wide variety of pharmacological classes and target biology. There's also a wide range of log P values, measurement of hydrophobicity of the compound, as well as molecular weight. So we felt that we had a pretty good, diverse test set to work with in this study. And the next slide presents the summary of results of the inter-laboratory assessment. And overall, between 60 and 70% overall concordance was achieved between each respective, which was a little bit disappointing to us, but it gave us an opportunity to learn a little more about what we saw in the data. So moving on to the next slide, what we found was the results agreed at a rate of 75 to 100% between the four laboratories. So in 19 out of 20 compounds, we had just about every lab agreeing in terms of their final stratagenic classification. Either they all agreed or 73 out of 4 agreed. But metoclopramide was one compound where there was sort of an equivocal readout between the four labs. And it was interesting because it was the two pondfish labs had the same classification, whereas the two main labs had the different classification in that case. In terms of cases where all labs had agreed on a certain tragenic classification, we had 15% of the time they were misclassified by the labs, and 25% of the time the compounds were correctly classified by the labs. We also learned during the course of this initial study that precipitation continues to be problematic, particularly at the high dose that was selected for the study, which was 1,000 micromolar, pretty high concentration. And even with the pH formulation work that we did, there was a real problem with running into pH shifts that were out of range of what was acceptable for culturing embryos without causing any adverse effects at these higher 1,000 micromolar concentrations. So we thought that could potentially influence the assay's overall performance. We also had some cases where all the labs either produced false positives or false negative classifications. And false positives included clozapine and loratadine, and a false negative one was fluconazole, an antifungal. Next slide. So we thought about different ways we could potentially improve and optimize the protocol. And one place was the thought that maybe including inter-experimental replicates might be a way to improve the results. And so various labs set out and took some individual experiments to look at the potential power of using replicate data. And this is just an example. And basically all of us came back and said, you know, replicates can correlate by 10 to 20%. And this is an example where one lab had tested all the non-tradagens. They had originally had a 60% concordance, but by using replicates, and it's frequency analysis is what's done at the end to get a final classification. So you ran something 3 times, and you had two called non-tradagens and one experiment and you would ultimately call it a non-tradagen because you got a call. And here, 80% concordance was achieved when frequency or replicate experiments. Next slide. Also went back and looked at whether log P had any correlation with their in vivo tradagenicity, 25 data, L25 meaning overall embryo toxicity, and we did not see any correlation whatsoever. So even though in theory one would think maybe a more hydrophobic or hydrophilic molecule could somehow influence overall toxicity properties, tradagenicity properties, there seemed to be no correlation whatsoever when we looked at these 20 compounds. Next slide. So some of our colleagues had some full expertise in the group, particularly from AstraZeneca. So they ran some experiments to see if the compounds that false positives and false negatives in everyone's had differences in embryo uptake. And it was quite interesting because they found that clozapine and loratadine had presented extremely high uptake values in embryonic tissue within 24 hours of treatment, whereas fluconazole had extremely low, less than 5% uptake. So there was a thought that something about an embryo's uptake of compound might influence the overall teratogenic classification in the screen. And there was some thought of perhaps permeating the chorion with an enzyme treatment might improve uptake because it'd be essentially punching big holes in the chorion, and maybe that would somehow rectify this problem. So we included that step in the optimized protocol. So moving on to the next slide, this is just a framework of some of the changes that were taken with the protocol optimization. So the framework of the protocol is the same as the original protocol, but then the concentration range was capped at 100 micromolar, pushing up to 1,000. And doing that, we could greatly simplify the formulation procedures and minimize all the pH adjustment work that we had done previously. And with that, we also were happy because there were cases in our data sets of compounds that are known to be unstable. And one is thalidomide, another one was dimethyldione that were included in that first test set. And there was some incidence of false negative data associated with those two compounds. So if there was a way to really streamline the formulation work and get that drug on the embryo as fast as possible, that might enhance teratogenic signal, and we actually found that it did. And we did some other things such as permeabilizing the chorion, including biological replicates in the protocol, and some places where we would adjust the concentration range. So for instance, if we ran the first experiment and there was a very steep lethality curve, we wouldn't necessarily run on a 0.1 to 100 concentration range in the second and third experiment. We would refine it based upon what we found with the lethality curve to try to adjust to that. So moving on to the next slide, we had a single lab run all 20 compounds again using the optimized protocol, and again these compounds were blinded, so the scientists didn't know what they were handling. And ultimately, this had originally had 60 overall concordance, achieved 85 concordance when they used the optimized protocol. So moving on to the next slide. So the final summary of phase 1 was our initial predictivity was lower than expected with the run, which was the 4-lab interlaboratory assessment. But then we learned a lot from that data set, and we worked together and put in a number of optimization steps in a refined protocol. And then that protocol was run again by one lab, and concordance improved substantially. So we ended this phase 1 at that point, and we moving on to the next slide, we just published the work, and that can be read in detail in this article on reproductive toxicology that I believe came out at the end of 2011 or early this year. Okay, so moving on to the next slide, I'll just talk briefly about phase 2, which is the phase that we're currently involved in. We took the refined protocol and ran the assessment with 38 blinded proprietary compounds, and these are compounds that 4 out of 5 of the pharmaceutical companies that are involved in the consortium essentially into a ring. So we wanted to get as diverse hemotypes as we could. So we wanted to get compounds. And those compounds were run at 2 separate labs. And what we did is we selected 2 labs that had the greatest discordance in the original phase 1 data. So I believe it was Lab B that used pond strain, pond fish, and I think it was Lab D, which used the proprietary strain fish. Those 2 labs only had about a 50% concordance in teratogenic classification in the first study. So what we're trying to do is really challenge the bar here by using those 2 very disparate labs for running this assessment. And we've completed all the experimental work, and we've completed all the cross-checking. So now we're going through the data carefully. Right now, we were happy to see greater agreement between the 2 labs. Now is about 86% agreement in their classification. And we're just getting to the point of getting all the concordance data in, because some of the rate-limiting sides of this, besides all the experimental work with all the replicates that's involved, is we also included compound uptake analysis on all 38 compounds. And in cases where there was less than 5% uptake into the embryo, that work was again repeated with a pushed up higher dose concentration. So it's been a bit of an extended evaluation, but we're hoping to be able to disclose that data very soon. So moving on to the last slide, the summary. I'll just say that from the work we've done and with some of the previous published work, we've concluded that the zebrafish embryo is promising as an animal alternative that could be used in developmental tox assessment, and studies, including the ones that the consortium are talking about, as well as ones that have been published, suggest good concordance for predicting in vivo teratogenic outcome using different designs of a zebrafish-based assay. So some of the future efforts that the consortium is interested in is perhaps working on a protocol that could achieve complete automation. And the big step here is going to be to find a way that the score system could be refined substantially, because that is very manual, it's very detail-based. If there's a way we could use statistical algorithms to perhaps cut it down to just a few critical endpoints, then maybe those could be applied to imaging work, where imaging algorithms could be put into place, and that could actually be all done in a high throughput fashion. And the other point is, again, overcoming compound solubility and uptake limitations. And we would like to eventually work together to find an alternative protocol in cases where we just cannot get compounds substantially in solution enough to run this type of assessment, and the microinjection approach will likely be the way to solve that particular problem in terms of still applying zebrafish for those types of compounds. So moving on to the next slide, I just thank everybody for their attention to the talk, and I'd be very happy to address any questions that people have.
Okay, thank you, Karen. Let's see, are there any questions from the attendees?
Okay, this is Akira Fukushima. Hello?
Yes.
And I have one question. I think that it's important to control the quality of water to apply for the fish.
Oh, I think it would be pretty important to have good water control. In our hands, we've used both DI and RO water for supporting colonies at different places where we've, different labs where we've had zebrafish. pH is extremely important in terms of being able to keep a very stable pH in the breeding facility. And when there's pH shifts, that can really affect not only fecundity of the breeding fish, but also the quality of the eggs. So a lot goes back to the actual husbandry side of having very good water quality in the tanks. In terms of what is used for the for making the fish media and so on, it seems this would be applied where you would want to be very careful about purity and water quality.
But do you think that it is possible to get rid of the agricultural chemicals just for the dialyzing method? I think that in order to get the high quality of water, we have to choose a kind of reverse osmolarity process. Don't you think so?
I didn't quite make out the last sentence that you said. So what was the last sentence you said?
Reverse osmolarity.
Oh,
very high quality of water is possible to get.
I would say it would be RO, water reverse osmosis.
Yes.
Yes.
What do you mean that dialysis is reverse osmolarity water?
I'm sorry, the molarity of the water?
No. You mentioned that water is dialysis, but is it reverse osmolarity water or not?
I think it's, he's asking if it's a river source.
Oh, I'm sorry.
Oh, yeah?
Actually, we've worked with a few different, we have one lab up in New Brunswick that I believe the water might be a river source. The water, we have a well water source at another lab, and the third lab, I believe, is from a lake, a reservoir. But it's considered extreme. I know that lake is considered extremely good water source. It's a watershed, highly environmentally clean a water source.
Thank you.
You're welcome.
Karen, this is Bruce Meyer.
Oh, hi, Bruce.
Hi, how are you? Very nice talk. Thank you.
Thank you. Thanks for calling
I have
in.
one question regarding, you have one parameter that measures lethality, right?
Yes.
And the embryotoxic portion, is that only on dysmorphogenesis, or does it also include lethality?
In terms of the morphologic assessment?
Yeah, that's only dysmorphogenesis, right?
Oh, okay. Yeah. So we first run a viability check against the concentration ranges, and that is our way of setting curve fitting for figuring out the LC25 concentration. But when it comes to dysmorphology, we do not morphologically assess embryos that are considered dead. So, and we have a set of criteria for defining a dead zebrafish embryo beyond just like no beating heart. There's other criteria. So if the embryo fits the criteria as being non-viable, they are not scored. So it'd only be viable embryos that would be assessed.
Okay. And will you make these slides available to the participants, or is everything covered in the publication?
Let's see. Well, I think there's no problem with making them available. I think that was something that Hesse was planning to do. A lot of the details, because so much of what I talked about today covers phase 1. That's in extensive detail in that publication that just came out earlier this year.
Okay. Thank you. Yeah,
You're
we've
welcome.
got a lab that's starting to work with zebrafish.
Oh, pardon me, I didn't hear you.
Oh, we have a lab that's starting to work with zebrafish, so I'll let them know about the publication.
Oh, good. Good luck with it.
Yep. Thank you.
You're welcome.
Okay, any other questions for Karen?
Hello, this is Jiro Seki from Astellas Farm, Japan.
Hello.
Yeah, hello. Thank you very much for a very clear and informative presentation. I'd like to know the current situation of the application of imaging technology for morphological analysis. So are you using some technology, imaging technology for morphological analysis?
At this point, no. We're just using visual assessment, basically evaluating through a stereo microscope and using the SCORE system. But there has been some really good breakthroughs in automated imaging technology. In the last year or two, there was a publication that was published by, I think it was Nature, by a MIT group that has seems to have cracked the bolt on the mysteries of how to image zebrafish at a high resolution in a high throughput fashion. Because the one thing about zebrafish that are really difficult is that they're round and they tilt a lot, and it's very hard to get them in position to a consistent position to do very careful imaging, especially if you're looking for subtle changes in size or shape or what have you. And this particular lab published on an approach where it was designed specifically for zebrafish, and it used also some kind of confocal microscopy and nanolasers, where they were also including some kind of high throughput microsurgery in the evaluations. I think they were doing like axonal trajectory and tracking neurons, and that was all done in a high throughput fashion. So, we did speak with that particular group of scientists, and what they're planning to do is generate another one of these systems that they published on that would be used for, I guess, fee-for-service. It'd be fee-for-service public access type of imaging, and that'll be something that should be rolling out of MIT, I guess, in the near future. And I think that they really have, they're really at the cutting edge of a lot of the automated imaging technology.
Thank you very much, Karen.
You're welcome.
Okay, any other questions for Karen?
Can I have a question?
Sure.
Yeah, I'm Minishina from Sun Planet. And I have a question regarding the fish strains. Did you use the same strains in the collaborative studies?
Did we use the same strain, and which study was that?
Yeah, in collaborative studies.
In the
4 laboratories.
Oh, in the 4 laboratories.
Yeah,
Sorry,
yeah.
there's static on the phone. No, we actually had 2 laboratories had the same strain, and then 2 laboratories had different strains. So 2 laboratories used this, like a classic, what you would call a classic wild type, right out of the pond. Same source, but it's the pond fish. And then the 2 other labs had, like, which strains are very well, I guess it's a kind of a workhorse ecotox strain. And this proprietary strain that the 4th lab worked with was just kind of out there as their own strain. But we wanted to look at some differences in strain just to get a sense of whether strain is a real concern, and whether there might be some fish wild types that are more robust in the assay versus others.
So you think that there are some difference in sensitivity with the chemicals between the strains.
Yes, I think so. Yeah. And, you know, just like I can't say much about what's coming down, but I will say I think there's been some strain differences that are observed in the current set of data that we're getting in phase 2. And I will say the one lab is actually taking the pond fish, because they're used to using their own cultivated in-house strain. They're now taking the pond fish and they're rerunning some of the experiments to see if they get different types of data than what they did, because it looks like some of the disparateness is like, yeah, it just seems like everything else is absolutely the same between the two labs, and so it's pointing to one potential difference, which would be the strain.